Hello, hello everyone. So today we are going to study the seed science topics. So this is also part of the syllabus, and it has been asked multiple times about genetics and the agronomical principles of the seed production, and also the public and private sectors how they are dealing with seed production. So let's understand few important points that you should always remember. See, when we talk about genetic principles, it means that the genetics or the seed, okay, how the seed genetics, okay, is affected by different factors of seed production and how it can like manipulated or it can change. The seed, though it is like fertilization of the parents A and B, we get F1 seeds. Still, there are many ways in which this seed can be contaminated. Okay, so the contamination is possible in this seed, and once it is contaminated, it will lead to what will happen? It will lead to decreased yield and other features. Okay, so now we will be studying how these genetic principles, which we should take care or we should remember, okay, to overcome the what do you say the contamination part. Okay, so the genetic purity of a uh, variety or trueness to its type deteriorates due to several factors during the production cycle. So first point, some points I've taken from the e-agri material also, some extra points they would have given. So like the seed production in adopted area, the area which they are doing, that may be influenced by different factors, okay, in a particular area, and whether it is from an approved seed source, from an registered seed source or from some farmers or from some other places. So that also can influence. So now we will get into this topic. So there is possibility of developmental variation. Developmental variation means what? So it is like we are growing the plants like in a particular environment, photo period, elevation, and all these things. So based on that, there is possibility that during the development there can be some variation of seed, and this may lead to a change in the seed genotype, some form. Okay, hence grow the variety in the areas they are naturally adapted. We need to grow in the areas they are naturally adapted. Like it is said, seed production in adopted areas. Adapted areas means the area for which we have targeted. Okay, for example, you have grown it for Andhra Pradesh. Like you have made that variety or that variety, okay, for the seed production for Andhra Pradesh. If you go and grow it in UP, then this may lead to developmental variation. Okay, it can be anything. Like it can be the epigenetic factors. Okay, you may not know what is epigenetic, but the thing is there is a lot of possibility of different ways in which this developmental variation can happen. Also, the second most important is mechanical mixtures. It can be mixed from other plants. You're growing in a field, and there may be some volunteer plant. Volunteer plants means in the last season you would have sown some plants, some seeds. So they may germinate in the next season, and they may contaminate the field. Okay, so that is also possible. And also using the same seed drill for the another variety. Okay, or some using some gunny bags and bags which are which can lead to the mixture. Okay, the third is the mutation. Okay, so this is not like very serious, but this is possible because when you're growing a crop, there is possibility of a seed like the mutation in the plant. Once the plant is mutated, maybe it can change the characters in the next generation. So mutation is also a very key factor, but not generally because it is like happening in 10 raised to minus 6, and that's why not much significant, but you should remember it. And also natural crossing, like as I said, so there may be some other field also nearby. Okay, from there it can happen natural crossing. That's why we say we should have isolation distance. Okay, we should have isolation distance to match that up. Okay, so source extent of contamination depends upon the magnitude of natural cross fertilization. Okay, in self-fertilization, it is not as serious unless it is a male sterile or grown in proximity with one another. Usually in self-pollination, not big issue because they are already self-pollinated. There will not be any cross-pollination except if it is very near. But in case of our cross-pollinated crops, it's an issue. Okay, there can be natural crossing and the variety may be changed. Okay, so another is the minor genetic variation. It can lead to like phenotypically uniform and homogeneous when released. When we release, there may be uniformity, but later it will change. Okay, during later production owing to selective elimination by the nature. Okay, because nature is also playing a role here. Okay, while selecting the crops and modifying it, so that also can lead to a change in the genetic structure. So that is minor genetic variation that is also possible. Okay, that's why we all always depend on the breeder's seed. Okay, for these minor variation which are preserved. So care during the maintenance of nucleus and breeder stock is necessary. Means at least we should maintain in our what do you say, breeder seed. Breeder seed means that is the primary seed. Okay, where the seed, the variety is bred. So that should be at least preserved. Then there is possibility of selected influence of some pest or disease. Okay, so there may be some influence of some pest and disease. For example, you would have seen in vegetatively propagated crop, easily this virus and fungus and other things may attack them, and it is propagated again and again every generation. Okay, so the genetics. So in this way, there will be there will be variations. Okay, and also seed production under strict disease-free condition is very important. Otherwise, because of disease also, there can be some genetic changes which can happen, and that may deteriorate the variety and decrease the yield. Okay, and also it depends on the technique of the plant breeder, what type of technique he is using. Okay, whether he is following the isolation distance. Okay, whether he is using the crossing format, how he is crossing the two varieties. Okay, and whether he is like disease and other factors also is taking into consideration. He is taking into consideration the isolation distance. All these things are also key, which you should remember. Just in 5 minutes, you're able to understand, and that much only you remember. Okay. So next is the agronomical principles. This is also very, very important and most repeated. Okay. What are the agronomical principles of seed production? Let me tell you, when they ask you about agronomical, just think like the seed you have, then what you will do with seed, the next what you will have, what will happen. Okay. Then in the field, what you will do? There will be pest and disease. Then in storage, then you will harvest. Okay. Then there will be seed will be preserved. So something like that. So from here starting, what is starting? We will select the land. From here, like this, you just remember you will be able to write. This is very easy. Okay. See, first is agroclimatic and location. First, you should see which is the climate which is set for this specific crop, and then choose that. Okay, based on temperature, relative humidity, and everything. Okay, and next is isolation. You should have the specific isolation, means isolation distance for meeting the seed certification standards are there. Okay, so if you maintain that isolation distance from particular crop, like 400 meter we have in case of maize. So like that we have for different crops. So you can just give an example everywhere to get that extra edge. Okay, and also in case of hybrid seed production, time isolation can be followed. Time isolation means in different types of planting. Okay, uh, because here in cross pollinated there is huge chance of cross pollination. Okay, sometimes we can do time isolation, yeah, sometimes you can do distance isolation. Okay, and also choose the variety based on how. Okay, based on which should be for that area and desirable attributes, purity, yield, and other characteristics. Okay, and also while you are sowing, you should know like you are sowing male and female in some particular ratio. For example, 4:6 or 6:2 in different crops. So that also we should remember in agronomical principles. Next is rogging. Rogging means removal of unwanted plants. Okay, what we will do, we should see the plant in the field, and if some plant, okay, some see for example, they are all normal. Okay, like this, they are normal. Suddenly one is like big one. Okay, this you can show. Then this you should rogue. Okay, because this is some off type. Rogue it out. Okay, so this is like roguing technique which you should follow. Okay, based on the texture, color, disease, malform, all these things you should remember. Then we should go for pollination. Okay, supplementary pollination by providing honeybees in the hive. Okay, we can also help keep some honeybee boxes so that they can pollinate easily. These all agronomical techniques you can follow. Next is weeding. Weeding is removal of the weeds. Okay, you can use the cultivation practices, but clean cultivation practices has to be followed. Here all you can add some examples also. Next is irrigation. Irrigate at the proper time and flowering and vegetative flowering and maturity. These are the like major stages you should irrigate so that there will be uniform seed and good crop stand. Okay, and also like it should be stopped 2 to 3 weeks before harvest so that the seed is harvested well. Okay, so those all these things you should see. And plant nutrition, the amount of nutrient which you want to use, whether you have to use like micronutrients you should use for a particular disease or for a particular crop. These recommendations we need to use, so that you should remember. Okay, and plant protection. Okay, means what you can do now here, anywhere something you can give like example in plant protection. Okay, like Phalaris minor. Okay, this is for example in weeds, I'm telling Phalaris minor. Okay, it will affect weeds, so we need to rogue it out. Something like that, examples which will attract the examiner. Plant protection also, you can give some example, like if there is some diseased plant like Karnal bunt is there. So those, if that is affected, then that plant has to be removed. Okay, then finally what we do, harvesting. Harvesting has to be done and then approval of seed field for certification. Okay, and seed yield and quality should be maintained. Okay, care should be given to mechanical injury and mixing of seeds during harvest should be there. Means proper care should be given that there is no injury or anything to the seed. Okay, then we have drying. Okay, then we take it, then we dry it, then finally we go for seed storage. Like then we should use this malathion and fumigation for storage area, because you would have studied in the storage chapter also. Okay, so these are the major things we should remember. Now what we studied, genetic principles, agronomical principles in a very short manner. Okay, so some of the things you can remember, like foundation certified, how much it is. In wheat it is self-pollinated, we usually have 3 3. Okay, and in case of some pulses and all, you can general write ragi, black gram, green gram, 20 and 10. Okay, foundation it will be more, and certified 10 it is there. Okay, the next generation of seed production, this is also very very important, which is generally you should know. Okay, and it is asked in the previous year questions. So we have majorly 5 classes of seeds, but registered class sometimes we don't use them. We don't use, because in India we don't have this class, but these are the major ones. Okay, breeder, foundation, certified, and truthfully labeled. What happens? Okay, in case of breeder seed, seed or vegetable procurement directly controlled by the originating or the sponsoring breeder or institute. The person who is breeding, the person who is crossing this first person owner, and golden yellow tag for breeder seed. Should remember. Next, which is this foundation seed. Okay, maintain specific genetic purity. This is like very high amount of genetic purity. Okay, it depends on crop, but up to 99% and all it is there. Okay, genetic purity. It can be 99, 99.5. Don't worry about that in some books. Okay, it is all nearby. And in like in white tag is given in case of foundation. Okay, in breeder seed, but we have 100%. Okay, completely pure. These seeds will be like 99%. Okay, registered seed, if you want, you can write progeny of foundation seed, the next generation of foundation seed, handled to maintain genetic purity and approved for seed certification. Okay, so this foundation seed, what happens, uh, this is like usually supervised by agriculture experiment stations. Okay, and also the persons involved like NSC. Okay, National Seed Corporation. Okay, and State Seed Corporation. All these things are also involved in case of foundation seed production. Next is the certified seed. Okay, this is the progeny of foundation seed, and it is produced to handle genetic identity and physical purity according to standards specified for the crop being. Okay, so here also like some progressive farmers also can be involved in this. Okay, we provide them seeds, and the farmers will grow them and then supply back to the seed company or the organizations. And then we have the truthfully labeled seeds. Okay, it is applicable to both notified and released varieties. Here, what we do now, only physical purity and germination tests are only done. Okay, only physical purity and germination tests. But if you go to through this all India coordinated research trials and all, here you have to go through all these procedures. Okay, this is a long procedure. But what happens in case of like mainly this is done by private seeds, private sector seeds. They go for truthfully labeled, means they label their physical purity and germination only. They will do that and then they will take in the market. Okay, but here also seed inspectors can take samples and check them. Okay, here the tag is, here I told you in case of certified seeds tag is azure blue. In truthfully labeled it is opal green. Okay, remember all these things. Just see breeder, golden yellow tag. Okay, breeder, golden yellow tag, foundation, white. Okay, certified, azure blue, truthfully labeled, opal green. So these are the stages also like this. We can make one diagram like this, like in the flowchart format. Okay, but truthfully labeled seed will not come in this. Okay, this is different. Okay, truthfully labeled seed, usually the private sector does, and directly only physical purity and germination is done, and they release it, they label it, and they release it. Okay. So what are the major procedures we carry out? Okay, in case of seed certification, we will do field inspection. Okay, I think you know what is field inspection. It is done by the officers. Okay, seed certification officers, they will be doing it. They will take something called field count. From the field, they will come and take the samples. Okay, they will check for physical contamination. Okay, maybe in some different crops are there, or some objectionable weed are there, like for example, in paddy, if Oryza fatua is there, some wild rice is there, then they will check that. Okay. And also they will check for some off types. Off types means which are outside the normal ones. Okay. Okay. Then we have like pollen shedders. Okay. Or shedding tassels. Like pollen shedders means uh, whichever are there. Okay, earlier only the pollen will start shedding in the female row. Okay, we have a female plot. Okay, female plot means only uh, we have a female plot like male sterile ones. Okay, we have a male sterile ones. Okay, that is female plot. Then here we have male one, but there are possibilities sometimes that here there may be some pollen shedders. Pollen shedders means they are not completely male sterile, or there may be some other issues by which this will produce pollen also. Okay, but this is female plant. Okay, then if it start producing pollen, then this all will be means contaminated, because here we are using male plot separately for pollination, but there may be pollen shedders. Okay, there may be some pollen shedders. Okay, so these are the important things you should remember. See, another important point I wanted to discuss is about public and private sector in seed production. This is also part of the syllabus, and these are also repeatedly asked. So here I will give you value addition. You know the basic things. See, there are more than 300 companies in India which are like private companies which are there. Okay, as per the Federation of Seed Industry, okay, the body, it says that about 18,000 crores is the private sector in India. That much worth it is. Okay, and we have like we are not exporting much, like less than 1,000 crores we are exporting, and we have lot of scope for improvement. Okay, and also we have varieties from ICR also. Okay, and private sector. Generally, what's happening is like in case of wheat. Okay, wheat, rice, okay, and pulses, sugarcane. All these things, our public sector is having upper hand, because what happens, these are high volume crops. Volume is more, but the value is very less. They don't have value, na. It will not be much profitable for the private sector. But what private sector does is they usually go for another thing is cross-pollinated crops, okay, which are high value crops. For example, tomato. Have you seen tomato seeds? Very small packet. It will be like in thousands. Few grams, okay, will be in thousands, 4 to 3,000 rupees. That much costly it is. Okay. But our public, such things they want to handle, private sector, not this rice, wheat, and other things. They don't handle much. Okay. And also presently what is happening is, you should remember, like we have a lot of MOUs. Okay, the private and public sector are having MOUs, and we are having a seed production MOUs by which both are combining with each other and doing. Okay, that is the status of present private sector. And and also like nearly 50-50 it is there. Okay, but now slowly the private sector is also gaining. Okay, in the seed industry side. And also some of the issues with our sector is like GM crops is there, na? they need approval. Like private sector will grow if we bring in more GM crops, then private sector will grow. Like we have Monsanto and other companies. So what happens is they have a lot of technology in like US, the companies in US, UK, everything. So if you give approval to GM crops, they will bring those technologies to India. But what happens, because GM is the major thing in those countries, uh, like they are working, like private sector is working. But what is happening is we are not giving approval. Okay, only cotton we are giving approval. So that's why the growth is not so fast. It's okay, okay, that's it. Okay. So if we have to suggest, then we should also give approval to GM crops by good analysis, and like the GSA protocol should be followed. So these are the things we should remember. So discuss 3, 4 topics in a very brief manner. Okay. Remember. Thank you.